Okay, good afternoon. Actually, good evening. I'm grateful to the organizers and especially my friend and president of EOPE, Dr. Yannis Bukovinas, for inviting me. It's really a pleasure to be here in this excellent congress and co-chair this session with my friend Dr. Nikos Ketoposidis. I'll pass the microphone to Dr. Ketoposidis to introduce our speaker.
Good evening from me. Mrs. Siravegna is going to give us a lecture about chronic evolution and drug resistance from cancer of avatars to liquid biopsies. She's sorry. She's postdoctoral research fellow in Cardiolo Cancer Institute, Italy. Please. Thank you.
Thank you. Good evening, everyone. Thanks for inviting me here. It's great to be here. And in the next half an hour, I will try to explain you what we have done so far for following clonal evolution in cancer patients, in particular in colorectal cancer patients, using many approaches, the in vitro and the in vivo models, as well as the liquid biopsies. So one of the major challenges that we have to face when we are managing cancer patients is the detection of early disease. So the screening of the healthy population, let's say, and that is very difficult to assess with regular imaging technologies. Also, we now know that cancer is highly heterogeneous, and we have to face the complexity of the molecular alteration among different patients and within the very same patient, the very same tumor. So the important thing and the most important challenge we have to face is also to identify the most suitable target to design therapeutic strategies that could enhance and prolong the overall survival of cancer patients. And in fact, now we have found a drug that are toxic sometimes and not beneficial at all, or maybe they are toxic but still beneficial. Maybe some drug combination are not toxic but even not beneficial, and others that are not toxic but beneficial, which is the optimal options, of course. So how to study the clonal evolution in these patients and how to get a real overview of the complexity of the disease? Well, we have set up in our laboratory in Italy an approach that is called the liquid biopsy. And when we refer to liquid biopsy, we usually refer to circulating tumor cells and circulating tumor DNA. So as you can see in the picture here, circulating free DNA, which is the only form of liquid biopsy I will talk about, is released into the blood and other body fluids through a mechanism of cell death and active release. So upon necrosis and apoptosis, these very short fragments of DNA are released into the circulation and can be isolated from plasma and serum, and several molecular alterations can be identified within these fragments. These type of analysis allow a real-time monitoring of patients in a non-invasive way, and it can be potentially cheaper than current biopsies. The difficult thing about the liquid biopsy is that the tumor content of the circulating free DNA is often limited, as compared to the normal DNA that is continuously shed by our cells in the body. And so the problem is to find that tumor-specific fragment, the so-called needle in a farm. So that's why we have implemented two different approaches, and in particular, we exploit very sensitive technologies. We have a single or multiple candidate gene analysis approach, and we use the beaming and the droplet digital PCR technologies, two digital-based PCR that are able to detect very low-frequency tumor alteration in the circulation, and in particular point mutation, short indels, copy number variations, and also we applied the next-generation sequencing technologies with the sequencing of the all exomes or a part of it with a panel of genes that we designed in our lab. So, which are the main clinical application of the liquid biopsies in the management of the cancer patients? Of course, at diagnosis, you can use a circulating free DNA analysis to genotype the circulating DNA directly in the blood to obtain a profile, a complete profile of the disease. And in combination with imaging, of course, you can identify the localization of the different metastases. At surgery, you can analyze circulating free DNA to understand whether the patient is disease-free or if he still has a minimal residual disease. So something that is very difficult to detect by the standard radiological imaging. Then you can use the ctDNA analysis to follow the patient through treatment and to monitor the patient in real time and to assess how the patient is following up, how the patient is responding to treatment, and eventually if he's progress with the emergence of resistance. And ultimately, at resistance, you can reanalyze the circulating free DNA in order to understand if there is any molecular alteration arising at resistance that can potentially be targeted with further therapeutic approaches. So the tumor that we are most focused on is colorectal cancer, that, as you might know, is the third most common type of cancer worldwide and the most lethal, and it slowly develops through various stages until it reaches the metastatic stage and metastasizes through the lymph nodes and through the normal mucosa in other organs such as liver and lung. There are several treatment options for this type of tumor. In the early stages, we have mostly surgery and then radiation together with standard chemotherapy, such as FOLFOX, FOLFIRI, and anti-angiogenic therapy, so the biological agents such as bevacizumab and regorafenib, and more recently, the anti-EGFR therapy and other molecular targeted therapies against other receptors which are altered in this disease that have improved the overall survival of these patients. Talking about blood, the only serum marker that we have in this disease is the CEA, the carcinoembryonic antigen, and as you will see, is not so useful in monitoring effectively in real time those patients. So, regarding liquid biopsies, I will now show you a very interesting study that we have started back in 2011 against the HER2 receptor, which is altered in 10 to 20% of colorectal patients. So in 2011, we conducted a study, and these results were first validated in the animal models, and then in 2012, we published the first study in Carton Discovery, and the first patient was treated according to our data at Niguarda Hospital in Milan. In 2013, we achieved the first partial response in this patient, and then in 2014, the actual clinical trial, which is called Heracles, started. So the trial was about HER2 amplified metastatic colorectal patient that resulted as, according to our in vivo models, to be sensitive to dual HER2 blockade with lapatinib and trastuzumab. As you can see from the graph here, while the trastuzumab alone and the lapatinib alone were not so effective in controlling the disease, the combination of the two actually achieved a major benefit in tumor shrinkage. So these are two examples of patients. In the first one, we have lung metastasis that responded very well to the dual block of HER2 receptor, and then another one with liver metastasis that achieved again a major partial response during treatment. So these are the results of the overall trial. In the waterfall plot, you can see how the vast majority of the patients controlled their disease with the dual treatment, and a lot of them also achieved a partial response, and we also had 2 complete responses, as also seen by the spaghetti plot, and 2 patients actually are still ongoing with the treatment with trastuzumab and lapatinib. So we are now moving to the next step. We are studying the acquisition of resistance in this patient and also the primary resistance. And you can see here in this graph how liquid biopsy helped us in uncovering the molecular alteration that was responsible for the acquisition of resistance. You have plotted here the HER2 copies throughout the treatment. These are the blood draw that we obtain every 2 weeks. And ctDNA analysis uncover the presence of PI3 kinase mutation that went up in the circulation only at progression and was absolutely absent in the baseline blood draw. So another target that we have been focused on for a while now is EGFR. This receptor is altered colorectal cancer patients tumor are addicted to this receptor, and the 2 drugs that were approved for the treatment of colorectal cancer, the anti-EGFR monoclonal antibodies cetuximab and panitumumab, are acting against the receptor because they are blocking the receptor, inhibiting its binding to the natural ligand, and then therefore blocking the dimerization and activation of the receptor, and therefore blocking also the RAF MAPK pathway and the PI3 kinase AKT pathway downstream. And so inhibiting the cell proliferation, the survival, and the uncontrolled migration in angiogenesis in this tumor. Unfortunately, not all patients can benefit from this treatment, and in fact, previous studies demonstrated that alteration in KRAS as well as RAF, that accounts for more than 50% of the patients, actually are refractory from treatment with cetuximab and panitumumab. And in fact, despite the blocking of the receptor, mutation in RAS and RAF keep active the pathway, therefore leading to uncontrolled cell proliferation and survival. But 15% of the patients actually do not carry any alteration in these genes, and that's why they are initially sensitive to treatment with cetuximab and panitumumab. But we have observed that unfortunately this sensitivity is only transient, and in fact eventually all patients after a period of 9 to 12 months, also when achieving a partial response, they eventually progress and acquire resistance. So when I started studying this problem, we knew that a patient acquired resistance had KRAS mutations in about 50% of the patient, but still many mechanisms were unknown. So we started following longitudinally this patient, and what we usually use in the blood is to track the 2 main alterations. The green line represents the APC mutation, so one mutation that we call a founder mutation because it occurs very early in the tumorigenesis of colorectal cancer, therefore it should be shared by all cancer cells. And then the KRAS mutations that is responsible for the acquisition of resistance. And you can clearly see how this dynamic of circulating free DNA mutation detection actually correlates very well with the clinical outcome as observed by the standard radiological imaging. And most importantly, it often anticipates the emergence of resistance of several months as compared to the radiological imaging. But then we went on and we wanted to discover many other alterations that could be responsible for the acquisition of resistance. So we selected nearly 20 patients for which we collected blood at baseline, so prior to anti-EGFR treatment and then at progression after a stabilization of a partial response. And we again confirmed the presence of many KRAS mutations as already known, but also we identified many actionable targets, such for example MET amplifications that can be targeted, for example, with crizotinib, and also novel EGFR cellular domain mutations that I will talk about later. So summarizing, the main mechanism of resistance to anti-EGFR therapy are the mutations in the downstream pathway, so mutations in KRAS as well as NRAS and BRAF, and also mutations in MEK. And all these mutations were validated in independent cell models that we generated in our lab. And all these mutations taken together accounts for approximately 70% of the patients. Also, more recently, we discovered the presence of these EGFR cellular domain mutations in collaboration with Clara Montagut in Barcelona. Okay. So now I would like to show you three cases, three way in which we exploited the ctDNA in clinical practice. And in particular, I will start from the use of liquid biopsy to design additional rounds of therapy. So speaking of the EGFR extracellular domain mutations, as I told you, these mutations often are acquired upon treatment with cetuximab and panitumumab, and these mutations inhibit the binding of the cetuximab of panitumumab to the receptor. That's why there are other drugs that have been designed, for example, the MM151. It is a trivalent monoclonal antibody that is able to bind the EGF receptor in different portions where the mutations are not affecting the binding. And patients were enrolled in clinical trial with this MM151 treatment, and we had the chance to follow the patients throughout the treatment in the trial. And again, you can see how liquid biopsy clearly correlated with the clinical outcome of the patient, and the blue line is representing the circulating EGFR extracellular domain mutations, while the gray bars represents the tumor burden. And again, eventually this patient responded very well to the targeted treatment, but then progressed. Then we wanted to understand whether the ctDNA analysis could be used to assess a lesion-specific response to a given therapy. And I will present to you here a very interesting case that we followed in collaboration with Dr. Corcoran in Boston at the Massachusetts General Hospital. And this patient was a metastatic colorectal cancer patient that had has been treated with several rounds of treatment, both standard chemo, as you can see, oxaliplatin, 5-FU, irinotecan, and then was challenged many times with anti-EGFR therapy, always achieving a good response. But then eventually, in late 2014, progressed to cetuximab treatment. So at progression, the patient presented many liver metastases, and only one of them was obtained by regular tissue biopsies. So we sequenced all the tumor specimens that we had available, and we performed next-generation sequencing of the primary tumor, of the liver metastases obtained prior to anti-EGFR treatment, and one of the many liver metastases obtained at progression. And as you can see, we detected the presence of the very same founder mutation, the TP53 mutation, occurring very early in the tumorigenesis. And then while we could not find any RAS or RAF mutation explaining the acquisition of resistance, only in the metastases obtained at progression to the last round of anti-EGFR therapy, we uncovered the presence of a novel MAP2K1 mutation. So we took advantage of the cell line bank of the resistant cell lines that we have generated in the lab. So starting from a sensitive cell line to cetuximab or tranitumab, we continuously treat those cells until they acquired resistance. And we have profiled all these models that recapitulates very well all the mechanisms of resistance that have been found also in patients, so KRAS mutations, NRAS, BRAF, as well as EGFR cellular domain mutation. But most importantly, in this case, we also had a model that carried the very same mutation of the patient that I previously shown. So we performed a pharmacological screening using this model, and we found that while these cell lines was absolutely resistant to either panitumumab or cetuximab given in monotherapy, the cell lines were absolutely sensitive to the combination of anti-EGFR with the addition of a MEK inhibitor. So the patient was treated accordingly, and this is the waterfall plot summarizing the different responses of the several lesions that the patient had at the moment of the combinatorial treatment. So some of the lesions actually responded very well, and some other lesions only achieved a stable disease, and some others progressed right away. So we wanted to understand what was going on in this patient, and we exploited the liquid biopsy to understand its clonal evolution. And again here, using liquid biopsy, we tracked two different molecular alterations, the T53, the black line that represented tumor burden, and the MAPK specific mutations as a marker of tumor response. And while we clearly saw that the red line, so the MAP2K1 mutation, actually responded very well to the combinatorial treatment as predicted by our in vitro model, the T53 mutation showing tumor burden, while dropped quite rapidly after the starting of the treatment, it grew up very rapid as well during the treatment, although the patient was clinically responding. So we wanted to understand what other molecular mechanisms could be underlying this rapid acquisition of resistance, and we subjected the blood draw obtaining prior to treatment to next generation sequencing. And we confirmed the presence of the TP53 mutation as well as the MAP2K1 that was found in the tissue biopsies, but we also uncovered the presence of a KRAS mutation that was absolutely missed by the previous tissue biopsies. Of course, we follow longitudinally also these KRAS mutations, and you can see how it parallels very well the overall tumor burden of the patient, explaining that this patient was actually progressing upon this combinatorial treatment due to the presence of a KRAS mutations. So the patient agreed to a new second biopsy of one of the lesions that never responded to the combinatorial treatment. And we confirmed the presence of a KRAS mutation that was for the first time identified by liquid biopsy only. And again, in the lesion that responded very well to the treatment, only the MAP2K1 mutations was found. So this is a clear example of how clonal evolution was kind of shaped by the different treatment throughout the clinical story of the patient, and how only the liquid biopsy was able to recapitulate the complexity of the molecular heterogeneity in this patient. So the last example I would like to give you is how we have decided to exploit the ctDNA in clinical trials. So as I was saying, until now we have followed patients during the response to anti-EGFR treatment and other targeted therapies until the emergence of progressive disease. But we didn't know what happened upon progression. And so we started following patients after the withdrawal of the anti-EGFR treatment, and this is just an example. So the gray bars represents tumor burden, and the black line represents the KRAS mutation that was found only at acquisition of resistance, and then when the patient stopped treatment with anti-EGFR but was switched with another standard chemotherapeutic treatment such as FOLFIRI, we clearly saw that while the patient was still progressing to standard chemotherapies, the KRAS mutation rapidly dropped down in the circulation. So at the molecular level, the situation came back to the initial one prior to any anti-EGFR treatment. So we wonder if a challenge therapy could be effective in these patients. And we had the chance to follow another interesting case. And this patient here was initially treated with irinotecan and cetuximab, and while he responded quite well, at progression he developed a KRAS mutation in codon 13. Then the patient was left off treatment for a while, and as you can see, the KRAS mutation went down in the circulation, and completely off-label, this patient was rechallenged with an anti-EGFR therapy with panitumumab. And again, you can see that while the patient responded quite well, the KRAS mutations went up again in the circulation together with a second clone. So based on these data, we designed the CHRONOS trial, which is an open-label, multi-center phase 2 trial, which stands for Rechallenge with Panitumumab, driven by RAS clonal-mediated dynamic of resistance. And in fact, we are planning to follow patients that are initially responding to a first line of anti-EGFR, and then when they acquired resistance, we will screen them directly in their blood to see whether they have acquired RAS mutations. Then we will follow serially in the blood the evolution of these mutations, and if the patient will show a drop in the mutational frequency of the RAS mutations by 50% or more, they will be rechallenged with the anti-EGFR treatment, and we will of course follow them during treatment every 15 days and eventually study the mechanism of acquisition of resistance to this second challenge of anti-EGFR. We will have the kickoff meeting next week, and the trial will start recruiting in September. So with this I conclude. Of course, I thank you all for your attention. This is the group I work with, and the supervisor is Professor Alberto Bardelli, and these are all our clinicians around the world and in Europe. Thanks to all.
Thank you very much, Dr. Siravegna, for sharing with us this very interesting data. Any questions from the audience? Yes, please.
Sure.
I was wondering, there are normal ways that the body fights cancer at presumably very low levels. I was wondering how far your liquid biopsy technique is from detecting the natural level which the body fights cancer.
Do you mean with the immune system? Okay, so the liquid biopsy has not been used extensively in assessing the evolution of the immune system. I can tell you that we are now working on measuring the neoantigen level in the circulation, but we are still talking about metastatic disease, so not very early stages. And a technology that we are now approaching is the sequencing of the T-cell receptor to predict the antigens that are exposed by the tumor and eventually fought by the treatment itself and by the immune system itself. But again, we are talking about metastatic disease and not early detection, unfortunately. We have not reached the sufficient sensitivity in the technologies that we use to be able to screen the healthy population of very early diseased patients. Yes, we also do methylation analysis by next generation sequencing and also candidate approach. Yes.
Other questions? Thank you very much,
Thank you.
Dr. Siravegna.
Okay.